professionals in the area of biomedical engineering. This platform serves as an opportunity to upgrade their knowledge on recent technologies. For the course of next 5 days, we welcome over 16 speakers from various organizations to enrich us with valuable information. The topics which will be covered, to name a few, it's machine learning in biomedical engineering, biosignal processing, precision healthcare technologies, design and calibration of medical devices, wearable devices, etc. We have had registrations from about 142 participants from different domains such as research scholars, medical professionals, academicians, and industrialists who will be joining us in the journey of gaining knowledge. We hope this event will become a successful one with active participation from all the attendees. We welcome you all for the event and wishing you all a happy learning. I would like to invite our HOD, Dr. A. Kavida, to share few words with us. Over to you, ma'am.
Thank you, madam. Am I audible?
Yes, ma'am, you are audible.
Thank you. So, I wish all the very best to the coordinators of this event, and I'm very happy to see the participants' numbers. It's you said it's 142. It's really an interesting count. Um, the very topic, precision healthcare technology is brought in because SSN College of Engineering in its 2021 regulation is planning to offer honors or say specialization in precision healthcare technology for the BTech students. So the BTech students joining in 2021 will have a new curriculum and the syllabi of the curriculum will include specific electives. Upon choosing those electives and graduating with good grades in those electives, a student will be entitled to have a honors in specialization of precision healthcare technology. With that goal in mind, we wanted to actually go in for what is new in this area. Now that everyone works in biomedical engineering, it's very interesting to know how to be more close with precision healthcare and how to see the various angles of precision healthcare in depth. So this is just a preliminary trial of finding out how to get in close with the precision healthcare and therefore widen our knowledge in the domain mentioned. So I hope it is going to be an interesting event because given the list of speakers, I'm really thrilled to listen to all the lectures and I hope you also will be. I don't want to take much of your time because we have very good speakers waiting to deliver their knowledge and I would like to get enlightened with the same as you are. So I wish the team and I wish all the participants all the very best and congratulations for hosting such a wonderful faculty development program in a good area like this. All the very best. Thank you.
Thank you, ma'am.
Thank you so much, ma'am. The first speaker for today is Dr. Abhijit Rajan. He is currently working in a medical device company called Boston Scientific as a senior scientist. He has been working there for about 1.5 years. The current job involves developing algorithms for detecting cardiac arrhythmia and cardiac implants, and previously he has also worked as an algorithm engineer for Philips Healthcare. He has completed his PhD and Master's from the University of Florida. In neuroscience attention in humans using EEG and fMRI data. He has more than 8 years of experience in biomedical engineering working on topics including biosignal processing, machine learning, statistical analysis, cognitive neuroscience, and cardiac arrhythmia. A hearty welcome to you, sir.
Thanks a lot for the introduction. I hope you can hear me. Just wanted to check.
Yeah, you are audible, Abhijit.
Thank you. So just give me a minute. I'll present my screen. This is the first time I'm using this software, so pardon if I have any delays here. So can you see my PowerPoint slides?
Yes, sir, it's
Yes, sir.
visible.
Yes, sir.
Visible,
Okay,
sir.
great. Okay, great. Oh, just a minute. I would just place the call in another screen. Okay, thanks a lot for the introduction and really honored to present here. Thanks for inviting me, giving me this opportunity. Um, so I'm Abhijit. I'm right now in Minnesota, USA. I'm working for Boston Scientific as a senior scientist. So to give you some background on my experience, I did my BE from SSN College Biomedical Engineering not so long ago. So this is a great opportunity for me to come back and present my research. Um, and then later on, I did my PhD in University of Florida in biomedical engineering, and my thesis was in cognitive neuroscience. So I used the EEG signals, fMRI, functional magnetic resonance imaging signals, and eye tracking signals to analyze various cognitive functions in the brain. And later on, I moved to industry, and I ended up in Boston Scientific as a senior scientist, and I'm working on cardiac implants. I'm working on the algorithms that goes inside some of the diagnostic and therapeutic cardiac implants. So um, I don't consider myself a thorough expert in any particular domain, but I had various experiences in different kinds of biomedical research like biosignal processing, machine learning, statistical analysis, cognitive neuroscience, working with different cardiac arrhythmias and algorithms related to cardiac arrhythmias, and medical device industry R&D. So my goal is to just kind of give you some highlights on some of the projects I've worked with, and hopefully this could be of some use to you. And please like feel free to interrupt me or ask me any questions at any point. And I would, it will be great if this is a discussion. Like it's a great even learning opportunity for me from based on some of your questions. So feel free to stop me at any time you have any questions. So I'll just talk about some basics here. What are biosignals? So some of you might already be well versed with all these basics, but just for someone who is not familiar with this, so basically biosignals, we are measuring physiological activity which can be measured and analyzed over time. It's commonly electrical signal, but you can also measure some non-electrical signals. So typically you have a biosensor that is converting your biosignal to an electrical signal, and then you have some filtering and pre-processing and amplifying that goes to kind of remove some of the unwanted aspects of the signal, just remove some of the basic noise from the signal. And then you store or transfer it. You either do like a wireless transfer like a telemetry, or you can do a wire transfer to a typically a computer or a display device where you analyze this biosignals. You can display it in a particular way you want to display, or you can do all kinds of analysis using various softwares. um, so some common examples, some of the biosignals are ECG electrocardiogram, EEG, EMG electromyograms, galvanic skin response. This is measuring the skin impedance. um, and you also have like some non electrical biosignals like pupil size measurement. So this is measuring your size of your pupil, which is a very exciting research field, and body temperature, things like this. And even this diagram shows you like all kinds of other signals like speech signal you can measure, and PPG, respiratory signal. So and each of these signals, there's some promising research going on. So my experience has mostly been in EEG, and ECG, and some of pupil size, and also fMRI. So I'm going to focus on those signals here. So the first biosignal that I was exposed to is EEG. So what is EEG, right? So we have seen this pretty often. Like these are basically recording electrical signals from the scalp. So you have an electrode placed on the scalp, and you are measuring the electrical signals that are generated by synchronized activity of broad collection of neurons. So when you place an electrode on the scalp, you are measuring kind of like a volume conduction through the tissue to the neural tissue, and that you're picking up on the scalp. So um, EEG is having a very good time resolution, so you can sample your EEG signal at a very high frequency in milliseconds, but it has a poor spatial resolution. The problem with that is that it's very difficult to localize exactly where the signals are coming from. So that's a slight limitation to EEG. So when you see electrical activity in a particular electrode, it's very difficult to pinpoint in like millimeters exactly which tissue, which brain region it is coming from, because a lot of synchronized activity from multiple brain region can be picked up in a single electrode. But now there are some exciting methods that can do source localization, which can kind of find the source of these electrical signals. But I think overall, I would say it is safe to consider that it has a poor spatial resolution, EEG, but it has a very good time resolution. Typically, we record EEG at a sampling frequency of 128 to 1000 hertz, and 1 to 40 hertz of your EEG signal can be useful. And so I would say, like, beyond 40 hertz, there is a lot of noise due to muscle movement and motion artifacts that gets added to the EEG. So it's difficult to analyze beyond 40 hertz, but still some people choose to analyze that. It has various applications, EEG, clinical applications. So one of the very common ones is detect epileptic activity. People go through like long-term EEG monitoring, like who have suspected seizure or epileptic activity, and they look for spikes in your EEG, and that can predict upcoming a seizure episode, or it can be used to detect ADHD, which is a attention deficit hyperactivity disorder. This is still, I would say, this is still quite a ongoing research. I don't think that this is a well-established clinical application, but in terms of research, it has like thousands of papers, I'm guessing, would be published on EEG. You use EEG to understand basic cognitive process like attention, working memory, cognitive fatigue, which we'll talk about in the coming slides. So a lot of research application, and very interestingly, there is some commercial application too recently. It's called neuromarketing. So this is a very novel and exciting field where people are using EEG to understand the customer responses to certain advertisements. So like it's kind of understanding how much does an advertisement or a marketing material, how much of an impact does it have in people's minds. So what is the emotional impact, and how much do they recall that advertisement, how much does this advertisement actually motivate them to buy this product. So that's they're using EEG and all kinds of fMRI, even fMRI, to do this. It's a very new and exciting field. Okay, so my experience has been mostly in the research applications of EEG, where I'm studying different cognitive processes through EEG. So one of the cognitive process I've studied is attention. So attention is such an important brain phenomena, right? So even I'm giving this presentation, and we need attention to even focus on the information that's presented to us. So we have to focus on the relevant information and discard the irrelevant. So that's basically what attention is. And I used EEG to study attention, and I wanted to give you like some kind of a basic result, or I would say like a very well-established result in EEG is looking at the alpha waves during attention. So alpha waves. So what is an alpha wave? It's in the range of 8 to 12 hertz in the EEG. Typically, I would say it's at 10 hertz. So if you take this signal from 0 to 1 seconds, you would see 10 cycles in 1 second. And it's very visible in EEG. If somebody has a very clear alpha wave, you can typically we see the alpha wave if you have a reasonable EEG measurement. So what that so in my research, the way the protocol works is that you are attending to a particular location in a visual field. So think of this as a video game. So it's like a subject is looking at the screen, and he hears a voice which says attend left, or he hears a voice which says attend right. So if he hears attend left, he has to focus on the left side of the screen and respond what is the orientation of the rectangle on the left side. So as you can see here, like there are two orientation of the rectangle. One is horizontal, and another is vertical. So if he heard the sound attend left, he has to respond like something like H in the keyboard to say that it's a horizontal. And then if we heard attend right, the subject has to respond V because it's a vertical orientation. So it's and these events are happening very fast. So think of it like every 3 seconds, the subject is hearing attend left or attend right, and they have to focus on that particular field. So a very interesting result in attention is that when you pay attention to the left side of the screen, actually your right posterior alpha power decreases. So if you see here, like the blue spots that you see, so this diagram is showing you the alpha power plotted for all the channels of EEG. And if you see here, for the condition where the subject is paying attention to the right side, to the left side, I'm sorry, his right hemisphere alpha power is much lower than the left hemisphere alpha power. And this kind of contralateral alpha effect is a very well established phenomena in attention literature. So the decrease in your alpha power is kind of indexing the level of your attention, and also the kind of the lateralization of alpha. So if you pay attention to the left side, your right alpha goes down, but your left side alpha goes up. And this kind of lateralization effect can be used to even study how well the subjects are focusing attention and how well their visual cortex is responding to the environmental stimuli. And attend right, you see the opposite phenomena of alpha. The left side alpha goes down and the right side goes up. Um, okay, let me know if you have any questions. I'll stop in between like for a couple of seconds and just see if there are any questions. Okay. So let's go to the next slide. So here, so the previous slide we looked at how we can use alpha waves in the EEG to study attention. Here, I'm talking about the theta wave in EEG, which is 4 to 8 hertz. So this is slightly lower frequency waves than alpha wave. So theta waves, typically people study theta waves for sleep. I mean, when you are going to sleep, your lower frequency power in EEG increases. But also theta wave is studied for higher cognitive processes like decision making. So theta wave is considered to be active during high cognitive control tasks like decision making. So how did we study decision making? Like, so as I said that there are two conditions, right? Attend left and attend right. And we added another condition which says choose. So the subject can actually choose between the two sides which he wants to attend to. So the choose condition is called willed attention, or it's a decision making, and the other two, attend right and left, are instructed attention. So um, here I have plotted a time frequency plot for the choose versus instructed condition. So this is contrasting the time frequency analysis for the choose condition with respect to the instructed. So as you can see at around, so here 0 here means like that's where the subject is hearing the sound. The subject has heard either attend left or he has heard choose. So after the subject hears the sound, you can see that at around 500 milliseconds, there is an increase in power in about 4 to 7 hertz, or you can say 3 to 7 hertz. So this is a typical theta wave increase. This shows that when you contrast the choose versus instructed condition and you look at the EEG signals and you contrast them and you do a power spectral analysis, you find that the theta power is enhanced during decision making. So this is specifically increased in the frontal channels. As you can see here in the left side, the frontal, central frontal. So frontal means like front channels, and center frontal channels usually have higher theta power. Um, so um, so I would say this is kind of a research application, right? So there is not like an immediate clinical application to this, but I think this kind of research makes you understand how does decision making work in the brain. Like what kind of waves do we see during decision making, and what kind of signals do we see from EEG. So that's kind of the purpose of this research. Okay, so here I'm talking about another application of alpha waves. So as I said earlier, we can use theta waves to study decision making. Here I'm using a computation called alpha lateralization index to study attention and decision making. So what is alpha lateralization index? So it's basically when you're attending left. As I said, if you attend left, your right side alpha goes down. So your left side alpha will go up. So you subtract your left side alpha with your right side alpha, and then you kind of divide the total power, the left plus right. So this number is if it's positive, then you can say that the person is paying attention to the right side. And if this number is negative, then then it could signify that the person is paying attention to the opposite side, because his alpha power is kind of showing an opposite effect. So we compute this alpha lateralization index at the time of decision making. So you can see here in the case of choose, the person has to choose between one of the two sides to attend, which side they want to attend. So here I have plotted a graph showing like the time series of the instructed versus willed condition, the alpha lateralization time series for willed versus instructed condition. So willed is meaning decision making. So you can see that the blue line is peaking at much earlier than the red line. So the blue, why is the blue line peaking earlier, is because when we are instructed to attend, our alpha lateralizes pretty quickly, because we don't have to do additional computation. We just are told, hey, attend left. So you just automatically lateralize your alpha to the left side. You pay attention to the left side, and then if you are asked to choose, the alpha is lateralized pretty late, like at least 400 milliseconds late, and that's because that's the additional kind of decision making that the subject makes. And at around 1200 milliseconds, at about 1.2 seconds, the subject has lateralized his alpha waves. um, Yeah, so this is again a kind of a research application here, trying to study what are the neural dynamics of attention, like how fast are we making these decisions, and what kind of neural rhythms are getting activated when we make these decisions. So if any of you are interested in going into much more details of this research on alpha and theta waves in decision making, you can look into this paper. Yeah, and please let me know if you have any questions on this. Okay, so the next application I wanted to talk about is how can we use brain connectivity? How can we do brain connectivity analysis using EEG? So, as you know that EEG is recorded from multiple channels, so we can actually look at the communication between two channels, or in other words, communication between brain areas. Um, we can use this measure called the coherence, or Granger causality. So coherence is telling you whether two time series, are they having similar, are they having high correlation in the same frequency band or not. So you can think of it as a correlation in frequency band coherence. And Granger causality is going one step further of coherence. So that is actually trying to claim whether one channel is causing the other channel. So it has this causality added to it. So I don't want to get into too many details, but I think if you're really interested in learning about the Granger causality, it is a very fascinating field where people use this method to understand the causal inference of one channel with the other channel. So does it mean, so they're basically trying to understand if one brain region is causing the activity in the other brain region. So you can see here that so I'm looking at the coherence in theta band, and the red bars are showing for the decision making, and the blue ones are the ones where we are instructed. So you can see that the coherence during this decision making is high, and also the frontal to parietal and the parietal to frontal influences are higher. So what does this mean? This means that during decision making, there is more communication going between our frontal and parietal regions of the brain, or in other words, like the anterior and posterior regions of the brain are communicating more during decision making. And this is kind of understandable also intuitively, right? Like if we are instructed to do something, we just kind of do it autopilot, but if I ask you to choose, then you kind of have to do additional computation, so you have to kind of recruit multiple brain regions to make that decision. And that's kind of what this is showing. Okay, so yeah, so I was done with EEG. Now I'm moving to fMRI signals. Um, so fMRI, this is also a fascinating research field. This is measuring the hemodynamic activity. So fMRI is not neural activity. It's not direct neural activity. EEG is direct neural activity, but MRI is measuring the changes in your blood oxygen concentration. So the the basic premise here is that increase in neural activity is resulting in increase in demand for oxygen. So, so when a region has more neural activity, it needs more oxygen. So this change in oxygen content is picked up by MRI, and it kind of tells you that this particular region is more active. So um, there is some kind of debate about does MRI and neural activity correlate with each other. I think they do to a great extent, but it is important to keep in mind that whatever conclusions we come up with MRI, we have to kind of have that caveat that it is not a direct neural activity of the brain, it's a hemodynamic activity. And but the greatest advantage with fMRI, which EEG doesn't have, is the spatial resolution. So you have spatial resolution in millimeters, so you can precisely understand which regions of the brain are more activated for a particular task. uh, and um, I would say, yeah, typically the temporal resolution is pretty low for fMRI. Um, it's usually in seconds. So as you can see here, the diagram shows that probably in 20 seconds, it takes about 10 seconds for the hemodynamic response to peak and come back. So it's a very slow biosignal, which is opposite to EEG, which is like extremely fast and um, high fluctuating. Um, Yeah, this is a kind of a typical result in fMRI, right? So fMRI is heavily used to understand and localize which brain regions are involved in a particular cognitive function. So here, so as I showed you previously about decision making, I was showing you like which neural rhythms are active, like alpha and theta waves. Now I'm trying to show where exactly are these decision making and instruction happening in the brain, which regions are more active. So if you look at the plot C, this is a contrast of willed versus instructed. So you can precisely kind of find out like which regions are more involved in decision making, and that's these regions called DACC, DLPFC. I mean, I don't want to get into details of this, but this DLPFC and IPL, this is kind of called the executive network of the brain. So as we have executives who are taking in corporates who are taking kind of high level decisions, so the same analogy they have used in the brain, where you have executive network who are taking kind of so-called involved in higher order cognitions. Okay, so the next application is to study brain networks involved in cognitive operations. So fMRI has been heavily used to understand some of the kind of brain networks involved in different tasks. So one very popular network is a default mode network. So this has been a very kind of established result in fMRI, I would say. The default mode network, it actually like as the name suggests, this network is active when we are in kind of default mode. What is default mode? Default mode is like kind of a resting state. So if you are just not doing anything, you're like kind of mind wandering, then you would see this network being active. So this network is having is consists of region called the PCC, which is kind of in your posterior region, and then PCC is called posterior cingulate cortex, and MPFC is called medial prefrontal cortex. So these two regions are heavily correlated. I've shown a kind of a example time series, so you can see from PCC and MPFC, and you can see like they're kind of moving up and down together, and they're correlated with each other. So um, these regions are correlated, and also they both are active during resting state. So this kind of network analysis is really fascinating, because now people are studying this network for even Alzheimer's disease and ADHD, even autism. And you find like this network is I think it's debatable, but my at least my understanding is that this network, if it goes heavily activated, that's a problem, and if it's weakly activated, that's also problematic. So it kind of has to be in kind of the sweet spot. But uh, it's a very ongoing research, and a lot of new papers are coming up about default mode network. I looked at the default mode network's main node called the PCC, posterior cingulate cortex, and um, so so I was more interested in attention. As I said, like this PCC and MPFC, they are more active during resting state and mind wandering. So our hypothesis was that when PCC is activated, then will that kind of impair the cognitive performance of a subject? So that's what we found that when so the y-axis here is the kind of activation levels of PCC, and you can see it's negative because usually this region is kind of going down during task. But if you see the blue bar, that is the case where the subjects are paying poor attention, and uh, it shows that during poor attention they were not able to decrease their PCC enough. So actually decreasing PCC is actually a good thing during task. So you have to decrease the activity in that region, but they were not able to do that enough. And that's kind of like our hypothesis here is that when we are doing high focus cognitive tasks, then we have this internal noise, kind of internal distraction, and that we are our hypothesis that is that that internal noise is coming from PCC. Um, and the second result is about understanding the networks during this internal distraction. So we found that there is actually during poor attention, we have less causal influence between DACC to PCC, but during increased attention, we actually have more causal influence. So this is based on the Granger causality analysis that I was talking about. It's looking at two time series and trying to find whether one time series is having a causal influence on the other one. So um, so it is kind of this research is the point of this research is to understand the networks and the regions involved during high focus and during less internal noise cases. So and people have even studied this PCC for ADHD kids, where you might have heard of this. Like some of the ADHD kids are not able to focus attention in one task, not able to complete it. So I think it's one of the hypotheses is that their PCC region is having some dysfunction, and the networks DMN, the default mode network, is having some dysfunction. So it's based on the same spirit. Just give me 1 minute. I'll have some water here. And please let me know if you have any questions. Okay. So the next topic is about machine learning in fMRI. So this is another very exciting topic here. So this is about using so if machine learning is basically like advanced statistical algorithms, right? We are finding the best model which can predict a certain, in this case, predict a cognitive state. So um, SVM is what the machine learning algorithm I've used. SVM is support vector machine. It's typically performing better than some of the classical classification algorithms. So SVM has been really popular in fMRI data analysis. So the way this machine learning works is we are trying to predict a cognitive state. So you can think of this in simple terms, you can think of this as mind reading. So can we look at a person's fMRI data and predict what that person was thinking? I'm kind of simplifying here, but just to kind of give you a high level view of what this means. So the task starts with asking the subject to think color or think space. So either you think of a color or like a red or green, or think of a spatial location, like think of left side or right side. So um, and then these are the two conditions, right? Color and space. And then we collect fMRI data over multiple time points, and each fMRI point is basically, you're basically getting a 3D volume every 2 seconds, right? That's what fMRI is. And now we look at a particular brain region, and then we kind of flatten that 3D volume, and we create a kind of 2D, a 2D, would you say, like a map, you can say that. And then where your columns is voxels, and voxel is, think of voxel as a 3D pixel, and your rows is trials. So say you have 100 trials. So you have 50 times you ask the subject to think color, and 50 times you ask the subject to think space, and you collected about, say, 1,000 voxels, 1,000 kind of points in the brain, and you have 100 cross 1,000 matrix. So once you have this 100 cross 1,000 matrix, you divide that to a training and test set. A training set is where you train your algorithm, you kind of find the best parameters of your algorithm, and then once you train that algorithm, you test it on the test data, and then you predict the labels and compare it with the true labels, and you get a decoding accuracy. So a high decoding accuracy in this case will tell you that we were successfully able to predict that the cognitive state. So why is this even important, right? So this is important because if the person is having distinct neural activity for these two conditions, then it will show up in your fMRI patterns also. It'll show up in your voxel patterns here. So, and the model will be able to pick it up. So think of it in this way, that if the subject was not paying attention, he was not actually following the instructions, and he was just like kind of mind wandering, then he would have no distinct activity for color or space, because he's like wandering for both the conditions. His mind is wandering for both the conditions. Um, and but if the subject did pay attention, then you should see a distinct neural activity in his voxel data. And that's kind of what we found here. So um, we looked at the decoding accuracy. So we have like 20 subjects that we did this research with. Out of those 20 subjects, we get 20, each subject we get a decoding accuracy, right, which tells you how well can we decode the thoughts, can we decode color versus space from his brain activity, from his fMRI activity. And um, the first thing that we found is that actually, if you look at the plot A, subjects with high decoding were actually having faster reaction time. So the x-axis here is the reaction time. The reaction time is like pressing the horizontal or vertical button that I was talking about. So it's so typically the subject takes about 1 second to reply, to respond, whether the rectangle was horizontal or vertical. Um, so if you see the red bars here, so this is a distribution of reaction times for subjects who had high machine learning decoding, and the subjects with low decoding had actually longer reaction time. So this is what I was hinting at, is that if you have longer reaction time, and if you have very slow reaction time, that means you probably were not paying attention to the tasks enough, and that could result in poor decoding of neural patterns from your fMRI. Um, and also we found that the so the B and the C plot is showing like the negative correlation between the decoding accuracy and the reaction time. So decoding accuracy is inversely related to reaction time, but also reaction time variability was low if you had high decoding accuracy. So which means that if you have distinct brain patterns, and you were also very consistent in your reaction time, you had less variability, and that's kind of shows in this. So um, uh, so why is this even important, right? So why should we think of this? What is the point of understanding this decoding accuracy? I think that there are some exciting fields in fMRI, like neurofeedback, where we decode patterns from a subject, and we show like a signal to a subject in the screen, and the subjects are able to kind of modulate their own brain activity through neurofeedback. So this has been this is a very active therapeutic research that goes for addiction or depression, where we are trying to decode a subject's brain activity, and we are presenting like a kind, say like a car, and the car is like the speed of the car is kind of the strength of the decoding of your brain activity. So if the person is trying hard enough, you would see the car is kind of moving faster. It's kind of giving a real time feedback of their brain activity. So I was not involved in that research, but I'm just giving an idea that this kind of research on the understanding the decoding of the brain activity can be used for such biofeedback mechanisms. Okay. Another exciting research is simultaneous EEG fMRI analysis. So, as I said, EEG is having very good time resolution, but poor spatial resolution, and fMRI is having good spatial resolution. So, if you record EEG and fMRI simultaneously, you get the best of both worlds. But it has a lot of challenges. This is like a picture of how this works. So you have a EEG cap placed on you, and you go inside this scanner. I mean, this is a huge magnet, and you go inside the scanner, and you are getting the neural activity, and also you are getting the hemodynamic activity from the fMRI um, data. So um, what are the challenges associated with this? So one of the biggest challenges that your EEG activity is corrupted with magnetic noise, or noise due to being inside a huge magnet. So there are like induced currents that gets added to induced voltages that get added to your EEG signal. But there are softwares that can filter out those MR noise, um, and I was able to use those softwares and able to kind of clean the data and use the EEG data, but it is challenging. But I think if you're looking at like low frequencies in EEG, I think it is possible to use simultaneous EEG fMRI. And here I've shown like an example of a research application of alpha bold correlation. So so the middle plot is showing you a correlation of the EEG alpha waves with the hemodynamic activity in IPS region. So, this is where I think this field is so interesting, is that when people study EEG and they look at alpha, they don't know exactly where that alpha is coming from, or where which structures are getting modulated with alpha. And if you combine the fMRI with EEG, you're able to study that. Now you can say that the alpha waves are kind of correlated with your IPS region, and IPS is called intraparietal sulcus, and which is a very important node in your attention network in your brain. Um, so this is kind of giving you the neural substrates of alpha. On the right side is you are looking at the correlation of theta with the uh, hemodynamic activity, the bold activity, and you see blue spots and one red spot. I mean, the blue spot is a inverse correlation, but the red spot is a positive correlation, and you see that the theta is positively correlated with the region called the DACC or MCC, which is a very important brain region that's for conflict processing. It's for making decisions. So this is kind of telling you more about where the theta in decision making is coming from. Okay. So now I wanted to move to ECG signals. These are much more easy to analyze. These are having much better signal to noise ratio than EEG. The amplitudes for this is much higher. That's in few microvolts, the EEG, and this is more in hundreds and 200s microvolt, maybe even more than that. Um, ECG is having very and some of you might be like much more familiar than me even. This is a very well established biosignal which has a very distinct morphology. Like there's something called the P wave and R wave and T wave. So P wave is in ECG is considered to be the activation of atrium, this the upper chamber in the heart, and the R wave is considered to be the activation of ventricles, the ventricular depolarization, and that's actually results in the contraction of the aorta in the heart, which sends blood to all parts of the body. And the T wave is like the relaxation phase of ventricles, the kind of the diastolus phase of the ventricle. So this QRS is very popular in saying that we need to detect the QRS in an ECG. That kind of tells you the heart rate. So by counting the number of R waves, you get the heart rate, because each R wave is like one kind of beat. You can imagine that, or one contraction of the left ventricle. ECG is used to diagnose several arrhythmias. These are some of the arrhythmias that we can diagnose with ECG. I'll talk about some of these arrhythmias in just a minute. As I said, it has much higher signal to noise ratio, and 3 to 40 hertz is typically the useful bandwidth. I'm sorry, this is a typo. This should be ECG. So that is 3 to 40 hertz is a useful bandwidth. Okay, so I had the opportunity to work on two devices that use ECG. Two implants basically that use ECG based detection. So one is on the left side is a diagnostic implant. This is a completely diagnostic cardiac implant. This is placed on your near your sternum. It's a very small device. It's 1 by 3 by 4 inches. This device is used for, so this is prescribed for patients with unexplained stroke or fainting or excessive palpitations. So if the physicians think that the cause of these issues can be due to cardiac abnormalities or cardiac rhythm abnormalities, now this device is being kind of prescribed. It's a very simple surgery. It's not like a highly invasive surgery. You're not doing like some kind of open heart surgery. This is just like on the skin, placing a small implant just underneath your skin. Um, this kind of implant is used to detect bradycardia, atrial fibrillation, ventricular tachycardia, and asystole. I'll talk about some of these rhythms. So this is a cardiac diagnostic device which sends all kinds of cardiac metrics, and it detects these important arrhythmia events and then sends it to the clinicians. And the second device that I got a chance to work on is called the implantable defibrillator. So defibrillator, like what is a defibrillator? Basically, defibrillator is sending a shock when there is a life-threatening arrhythmia. So in some of the movies, you would have seen this dramatic scene of a doctor shocking or a nurse shocking a patient, and that's basically defibrillation, right? You're shocking the heart so that it kind of there's a chance that it might restore to normal rhythm. The heart is pumping, the heart is having very abnormal electrical activity, so you have to apply that shock to kind of give a chance to get back to normal rhythm. um, so this defibrillator is prescribed for patients with sudden cardiac death, like with the risk of with the risk of sudden cardiac death, or like a structural birth defects, or a history of coronary diseases. So um, these are very specific patients that you prescribe this ICD device. Typically, I think this is one of the common symptoms is a myocardial infarction, which is like a loss of tissue in certain parts of the heart. And if your ejection fraction, which is kind of how much the ventricles is pumping blood out, if that's less than 30% or so, you prescribe this ICD device. So it's a therapeutic device because it is sending a shock inside your heart, and it's kind of trying to terminate life-threatening arrhythmias. So this device is mainly used to detect ventricular fibrillation. So let me talk about that. I'll talk about that in a couple of slides. Okay. So um, so what are the applications of ECG, right? One is a very important application is the atrial fibrillation detection. So this is part of the diagnostic device that I was talking about in the previous slide. So atrial fibrillation is when you do not have a P wave. You don't have a clear P wave in ECG. P wave, as I said, is an indication of atrium activity, atrium electrical activity. But in case of atrial fibrillation, the atrium is having a very chaotic activity, a chaotic electrical activity. So so that's why you see, if you see the wave ECG here for atrial fibrillation, it's it doesn't have a synchronized P wave because the atrium is just conducting in a very haphazard way. And this can be detected through algorithms. So there are very interesting and useful signal processing algorithms. Either you can do a single beat detection. So like for example, if you detect the R waves, then you go back and you define a window and you collect features in this window, like the slope of the signal or peak of the signal, and you could do a simple kind of linear regression kind of a logistic regression, or a simple linear regression to find out whether there is P wave or not. So that's a single beat detection. And also you can use like ensemble averaging. So ensemble averaging method is like averaging multiple beats of ECG and getting like one averaged signal. So if you do an ensemble average on a normal rhythm, you would see a P wave. But if you are kind of think of it as averaging 10 R waves put on top of each other, and then if you average those, you see a clear P wave. But if you do that averaging for atrial fibrillation, you see a flat line because the activity in the atrium is so chaotic that when you do the averaging, you'll get almost a number close to zero in that atrial time period. Um, the other important feature with atrial fibrillation is variability in RR interval. So if you see here, on the left side, you see normal sinus rhythm. So this is having a very consistent RR interval. But if you look at a AFib, atrial fibrillation, it has kind of this haphazard RR interval. That's because the atrium and ventricle are not in sync with each other. You are having like a very unexplained spontaneous contraction in ventricle, because the atrium is not having a proper P wave, which is like having a proper sync with the ventricles. So um, so this is another way to detect AFib. You detect the RR intervals, and you kind of calculate the variance of your RR intervals. If there is a high variance, you could say that there is more probability of AFib. So I'm kind of simplifying here, but there is a lot, many metrics you can use to quantify the variability. Variance is just one of them. So why is AFib even important, right? Why should we detect AFib? So one cause of concern with atrial fibrillation is stroke. So people who have chronic atrial fibrillation have more risk for stroke in the brain. So this kind of diagnostic device which detects this atrial fibrillation can uh, potentially save lives, or even it can prevent a, a stroke for a patient if we detect much before. So um, uh, the next clinical application here is ventricular fibrillation or ventricular tachycardia detection. This is the defibrillator device I was talking about. The defibrillator is detecting this ventricular fibrillation. So I spoke about atrial fibrillation, which is chaotic activity in the upper chamber of the heart. Now ventricular fibrillation is the chaotic activity in the lower chamber of the heart. So this is much more life-threatening, and this is much more serious than atrial fibrillation, because here the heart rate can go even above 200, and you see a ECG looking really chaotic due to ventricular fibrillation. And um, you see that there is no actually correlation with successive beats here, VF. This is called VF, ventricular fibrillation. But if you see a ECG of a normal patient, you see that the QRS complex are kind of having some correlation with successive beats. So this is a really chaotic activity that you see. Um, there are a lot of signal processing algorithms you can use to detect this ventricular fibrillation. So one is that you estimate the QRS complex. So as I said earlier in the previous slides, the QRS complex, you once you detect the R wave, you can look at the changes in your slope and kind of detect the Q point, and then also detect the S point, and then you can estimate the QRS width of your ECG. And um, once you measure the QRS width, you would find that for a VF, ventricular fibrillation, you have a higher QRS width, and your rate is also higher. And as I mentioned, you have poor correlation between successive beats. So these are some features that we use to detect ventricular fibrillation. And once we detect this ventricular fibrillation, we need to provide shock. So that's the shock delivery algorithm. So here is just a very almost like a cartoonish image here, but it is just trying to tell you that when you have a ventricular fibrillation, um, the device has detected that it has ventricular fibrillation, and then it's providing this shock, and that's the black vertical line you see. And after you provide the shock, you see a much more normal rhythm restoring. So this is considered a successful um, termination of VF using defibrillation shock. So um, so you can imagine here the time is very critical, because if a patient is in ventricular fibrillation for a long time, that can unfortunately cause a sudden cardiac death. Here, as soon as you detect that the patient is in ventricular fibrillation, you have to charge your capacitors and deliver the shock. So this is what the shock delivery algorithm is kind of trying to do. So it detects this VF using some of the features I discussed, and different devices have different features that they use, and I just provided some common features to use that. And um, once you detect that, you charge a capacitor, and you have to, once you apply the shock, then you have to also check whether the shock, whether it terminated your VF or not. So again, you have to kind of measure the rate and QRS complex and these kind of features to make that determination. Yeah, please let me know if you have any questions till this point. Okay.
Sir, do we have, hello, can you hear me,
Yeah,
sir?
yeah, yeah. Please, yeah.
Yeah. So as you said, after the atrial fibrillation, you are now checking with the normal ECG signal after
Mm-hmm.
the shock has been provided.
Mm-hmm.
Is there any specification that the ranges of the shock that has to be provided on demand of the atrial fibrillation? How much we have to provide, and
Yeah, yeah.
ranges?
So if I, yeah, the range is, it depends on the implantable device or whether it's a surface device. So for example, there are external defibrillators like the defibrillator you see in malls and police station where you kind of externally shock. There the shock is in joules. Is I think um, uh, I don't remember the exact value, but I think it's more than 40 joules in that range. So, and in the implant, also you have a range, a certain range of joules. I think it's in the range of 20 to 60 joules that you provide the shock with. And also for children, actually, you have a lower shock energy that you provide for pediatric patients. Um, I hope that answered the question.
Oh, thank you, thank you.
Thanks. Yeah. Um, so one of the key things for this VF detection is actually detecting the R waves. So because one key measurement is detecting the heart rate, right? Heart rate is coming from RR intervals. So the heart rate is telling you whether um, I mean, heart rate is kind of the first steps in the algorithm to determine if it's atrial fibrillation or ventricular fibrillation. So the very easy way to detect a R wave is using a simple threshold, like a 4 millivolt threshold here. Anything above 4 millivolt, you detect an R wave. Like any point you detect above that, that's the point of R wave. Um, and if you have a higher threshold, you miss the R wave, obviously, right? But if you have an extremely low threshold, you can detect a T wave. So what happens there is that you can have oversensing. So what does oversensing mean? It means that you are detecting R and T wave, and you are considering that as RR interval instead of detecting R waves and then next R wave. You're detecting like another wave in between. So you're actually double counting. So instead of actual heart rate being 100, you will end up estimating to be 200 because you are detecting this unwanted T waves in between. So this is so I'm giving a very basic method here, but the algorithms in the current devices are much more advanced than this. But still, it has the issue of T wave oversensing. That's what this is called, like detecting T waves. So this is a really exciting field of algorithm improvements where we are trying to reduce this T wave oversensing, because there are even like some very small percentage of patients where normal patients can get a shock, and that's really terrible, but it happens extremely rarely. But one of the reasons is like this T wave oversensing, where you detect the heart rate to be extremely high. um, and the worst case is that if you if you detect the T wave and you determine that this patient has to be shocked, the worst thing is to shock on the T wave. So if I showed you here diagram, like if this black point, if this was on the T wave, then you can actually induce ventricular fibrillation, and which is extremely undesirable effect. Like you are kind of inducing ventricular fibrillation on a normal patient by shocking inappropriately. Although this is extremely rare, but this is still an active area of improvement where we want to make that extremely low cases of algorithm. So the last signal that I wanted to talk about here is eye tracking. So this is a novel area of research. So this is using near infrared light and high resolution camera to track your eye movements and pupil size. So pupil size, think of it as a high resolution camera, and that is kind of tracking your pupil size changes, pupil dilation and constriction when you do a task. Um, so pupil dilation is a very reliable indicator of attention and cognitive effort. So when you pay more attention, when you are paying more effort, your pupil will dilate, and that's a common effect. But also pupil will constrict with visual stimulus. So if I flash a light, your pupil will constrict. So in cognitive tasks, it's very important to keep the luminance constant as much as possible, so that your pupil is not dilated to visual stimulus, sorry, constricted to visual stimulus. So we try to keep uh, the the lighting constant, because we are only trying to measure the cognitive effect of pupil, not the visual stimulation effects of pupil. So in my research, I basically looked at the pupil dilation in response to attention. So here, this graph is showing at 0 is where we present a kind of instruction to the subject, audio, which asks them to pay attention. And if you look at after 0, this y-axis is looking at the pupil dilation in percentage. So there is a small increase in pupil dilation. The pupil is dilating about 6% and about 7% increase in pupil size with attention. So another interesting phenomena is that if we look at the patients, look at the subjects who are performing better, who are having high performance results, like a high better focus and a faster reaction time in the tasks, their pupil were dilated slightly more than the low performing subject. So um, so this is a very exciting field, because pupil is completely non-invasive measurement. It's, you don't even need an electrode, you just need a camera to measure it. um, and it is kind of giving you a proxy measure of attention and effort. So I think although it is not giving a lot of detailed information, pupil, it's kind of only telling you if the person is paying attention or not. But it is still an exciting field to study human cognition, and especially study attention using pupil. And even you can think of in future, maybe you can have pupil based medical device for ADHD, or for some other attention deficits. You can try to use pupil to measure that. So pupil size, it varies with cognitive load. So cognitive load is like if I ask someone to do a heavy computation, a heavy mathematical computation, your pupil will dilate more. Um, Also emotional arousal. If you have exaggerated emotional response, like if I show you some very disturbing pictures, then your pupil will also dilate. It's an emotional reaction. Attention and mental imagery, like if I ask you to imagine certain images, very detailed images in your head, your pupil can dilate to that also. um, so it's a very fascinating field, but there is, but I don't know of any direct clinical applications to people right now. It's more in the stage of research. So I'm coming to my last slide here. So just a summary, these are the biosignals that I've worked with: EEG, fMRI, ECG, pupillometry. um, they all are having very exciting biomedical research applications. So I would say EEG is kind of picking up now. There is more, I see more and more, at least in US, I'm seeing more and more FDA approved devices using EEG. But still, I would say it is having a lot of challenges in terms of signal to noise ratio. So it's not, I don't think there are like well accepted therapy or diagnosis with EEG. Probably epilepsy seems like the only one where it has a direct application, but in all the other devices and clinical application, it's still, I would say, in the stage of kind of ongoing research and trying to make it more uh, established. It's still, there is some time to go before we start seeing EEG very commonly. fMRI is heavily a research tool, I would say, and there's not a lot of immediate clinical applications as I understand it, but there is a lot of upcoming research. You can Google biofeedback and fMRI feedback. That's an exciting field. ECG, as I said, there are, it has direct clinical applications. ECG, I mean, you can you literally save lives by doing signal processing on ECG, as I showed you, the ventricular fibrillation detection and atrial fibrillation detection also. um, but it is little bit, um, I would say, I don't want to say saturated, but it has, we need new ideas in ECG. I think maybe now machine learning and deep learning in ECG, that's a very exciting field to look at, and a lot of research on that. But there's a lot of work already done on ECG also, in terms of signal processing and getting like all the important features from ECG. And pupillometry is completely in research stage, I would say. And the methods I have used more frequently is digital filtering, power spectral analysis, cross correlation, SNR estimation, signal to noise estimation. I've done some machine learning analysis, classification regression analysis using logistic regression, support vector machine, um, decision trees. I wouldn't I haven't used a lot of deep neural network, and that's something I'm exploring now. I think one field that I'm just starting to learn more is about converting biosignals to images and using neural networks to do classification of diseases or different arrhythmia classification. So we can convert some of the time series to like a time frequency image, and we can do like a neural network based image classification on those time frequency images, or even um, there are other kinds of images like heart rate scatter images. So that's something I'm starting to learn, and it's a new field. I've used mainly Python, scikit-learn, NumPy, Pandas. Those are some of the tools, libraries I've used. It's a very, some of you might be experts in that, and if you are not new to if you're new to this, and if you want to learn more about biosignal processing and apply more, I would heavily suggest um, using these toolbox, learning more about it. And MATLAB is it's heavily used in research, and actually even in medical device industries I've seen people use MATLAB. It's also a great tool to learn it. And I think my first MATLAB was actually the first usage of MATLAB was actually in SSN college, and I've been using that for most of my projects here. So thank you. Thanks for your attention. Yeah, please let me know if you have any questions. I'll be happy to answer.
There are several questions in the chat box. The first question, which
Okay.
software, the first
Yeah.
question is which software predicts and gives this analysis? I think this was sent when you were explaining about EEG, sir.
Which software predicts and given this analysis? Yeah. So I've used like Python and MATLAB mainly, and I said I think these are the two main tools I've used: Python and MATLAB. Python, scikit-learn, NumPy, Pandas. These are some of the toolbox I've used, but um, yeah, but you have to do like some pre-processing, and that's kind of the initial steps. And then I would suggest MATLAB is really good for pre-processing, and it even has machine learning toolbox. So I recommend MATLAB. Um, Python, I don't have a lot of experience with the pre-processing part, but once I pre-process my signal, I think I use Python to do the final prediction. Okay, so how can we know QTC is normal or abnormal? Um, so if I understand, is it about the QRS, or I didn't understand this QTC? Um, I think if I understand correctly, if you're asking about the QRS or the
Yes, I think.
yeah. So it's the size of the QRS is it can tell you that if it is a wider QRS, it's abnormal. Like as I said, if you look at this if you just look at this image, right, like this is a normal QRS, which is much more shorter. So if you detect the R point, you can kind of go back and look at the changes in your ECG, and you can detect like the change through the changes in your slope. You can find the skew point, or you can find the point where it is crossing the zero line, and then you get the QRS width. If it is usually shorter width, I think 80 to 120 milliseconds is normal, and then beyond that you can get for VF you'll get a higher QRS width, so that's abnormal. Okay, sir, EEG database for Alzheimer disorder available? If so, where we can get it? um uh I don't know exactly which database for Alzheimer. Yeah, I'm sorry, I don't know exactly which database, but I will write it down actually, and I can get back to you on this. So let me just write it down. Yeah, maybe I'll send an email to the coordinators. I'll search and send you. Okay, how can I incorporate antennas into biomedical equipment practically? Um, yeah, I don't have like a direct experience with antennas. Um, yeah, sorry, that's not my expertise. Can you have resource person contact detail for teaching about biosensors? Yeah, so I can send you one good so somebody has asked me about an open source tool for teaching about biosensors. I have a couple of links that I can send the coordinators. I think let me write it down too. Yeah, that's a great question. In a ECG, can we have a resource person contact detail? Okay. Please let me know if you have more questions. Yeah, I'm sorry about the antenna question. I don't I don't have direct experience on that antennas. But if you have any other question, please feel free to tell me. I'll try my best to answer. Yeah. Prediction for Alzheimer. I have written it down. I'll try to find something and try to send the coordinators, and you can maybe or if you send me your email, I can try to send you directly. Okay, so I will. Uh, yeah, I'm just. I think yeah, there are open source database. Um, I need to check Alzheimer's, but there are surely open source database. I mean, I think some of the common ones that comes to mind is Kaggle is one. Kaggle. They have some data sets, neural data sets, but I'm not sure if they have Alzheimer's. But I can look into that and let you know. Let me know if I missed any other question.
There's one last question.
Yeah. Um, GitHub database. Yeah, probably GitHub could also have that. I haven't used GitHub much. There's also, I think that there's a Connectome project, if I'm not wrong, and that may have. Um, okay, so there's a question about uh, IMU measurement. Yeah, I'm sorry, I'm not directly familiar with that. Uh, so can you give me, so what exactly are you measuring there for arthritis?
Abhijit, can you hear me?
Yeah, yep.
I have one question. So,
Mm-hmm,
what Boston Scientific currently doing for this medical
mm-hmm.
kind of innovations? So, what
Yep.
is your current work going on, or what is the futuristic plan? What you are working related to cognition and all? Because yesterday I saw a TV ad given by Sony Corporation,
Mm-hmm,
like they are planning to release a cognitive intelligent based TV. So,
mm-hmm.
what your concern is doing currently?
Yeah. So actually, in Boston Scientific, I'm involved with cardiac implants. So I have moved from the neuroscience space, and I'm working on diagnostic implants and therapeutic implants. So the next phase of one of the next phase of Boston Scientific is using machine learning and deep learning to diagnose cardiac abnormalities. So what they are working on is, so if you think of the cardiac implant, it's the diagnostic implant. It is sending like it's sending clinicians metrics about their heart, and what we are trying to do is we are like trying to send those signals to the server first, and server we are doing like deep learning and machine learning, and then we are trying to estimate whether there is arrhythmia or not, and then we send that to the clinicians. So this is probably a long term project. So this is one of the things I'm working on. um, and also then other project is about defibrillator. So that is about improving reducing the inappropriate shocks in the defibrillator. So I'm working on some algorithm improvements so that we don't detect like abnormal rhythm on normal patients, and we don't shock normal patients. So I'm trying to reduce that percentage, working on the team with that. um, Regarding cognition, yeah, I'm more into now cardiac physiology. I think I'm not that involved with neuroscience directly. Okay, so
Thank you, Abhijit. Yeah.
right. So I think this mentioned that it's a measurement of joint angle. Yeah, I'm sorry, I'm not familiar with that. Um, yeah, sorry, I couldn't help much.
Okay, participants, do you have any questions you can ask, or else, yeah, you can go ahead.
Thank you so much, sir, for that very deep insight about a variety of biosignals. The next session will be at 11 o'clock, and I request all participants to join at 10:50. Thank you.
Abhijit, thanks for joining. Even in your late night, you taken such a nice presentation to us, and even you kickstarted this particular FTP. So thank you once again.
Thank you. Thanks a lot.
Thank you,
Uh,
Abhijit.
Thank you. Thanks a lot. It was great
Thank
presenting.
you, sir.
Participants, our next session will start by 10:50, and the session will be handled by Dr. Yasan Deepa, I think, who is a famous writer of the book Soft Computing Techniques. Um, uh, uh, Let us join for the next session by 10:50. Thank you all. Thank you, Abhijit.
Thank you.
Thank you, sir.
Thank you.